Now what we're going to study is photosynthesis. Photosynthesis is a topic that we've been studying since we were in the 6th or 7th class. What exactly is the word photosynthesis mean? When we're trying to understand the word photosynthesis, just briefing the initials, photo means light and synthesis for the synthesizing, any formation of something. When we're trying to understand the word photosynthesis, photosynthesis clearly makes us understand that the plants are trying to produce synthesized food with the help of photo, the light, and primarily given by the sun. Sun gives us the light, the plants are being provided the light by the sun, the plants try to use that light. The light comes in, plants use that light, and plants use that light to produce something which we call as food. And that is the thing, the food is trying, the food is what we eat, food is which keeps us alive, and that tries to keep the complete biodiversity in the complete earth. See, the word photosynthesis we just studied, detailings, if we get into the detailings, photosynthesis is done by the plants inside the green parts of the plant, the leaves. The primarily green parts of the plant, the leaves, and inside the leaves there is a cell of a leaf, mesophyll cell, and inside that mesophyll cell there is a particular organelle, and that organelle is termed as chloroplast. Inside the chloroplast, the photosynthesis is being done with the help of the chlorophyll molecule. This chlorophyll molecule traps the sunlight for the formation of the food. This formation of the food takes place in two different steps, light reaction and a dark reaction. When we understand the word light reaction, it is a reaction that should primarily be dependent on the light. That means when the sunlight is coming, the incoming sunlight has to be trapped during the light reaction, and this trapped sunlight is utilized to make certain products. And I'm trying to revise this thing again. This complete photosynthesis process we say are taking place in two different light and dark reaction. This light reaction is trapping the sunlight, and from this light reaction, after the trapping of the sunlight, two products are being formed, ATP and NADPH. These two products, the product of the light reaction, are utilized inside the dark reaction, and from this dark reaction we obtain the product, the final product of the photosynthesis, that is the glucose. Hence, this complete photosynthesis is taking place in two processes, light and dark, finally to produce the glucose. What we're going to discuss here now is the light reaction and how is this light reaction taking place. In this photosynthesis, the light reaction takes place. The light reaction takes place in two different steps, in two different type of steps. Light reaction taking place in two different type of steps, cyclic and non-cyclic. Cyclic and non-cyclic, two different steps. What we're going to study on the board now is we're going to study the non-cyclic process. Summing the complete thing, the complete non-cyclic process, in this non-cyclic process, the P680, which is the reaction center, part of the chlorophyll, along with the antenna molecules, absorb the sunlight, H mu, which is the sunlight. See, H mu sunlight is what we understand. We understand the chlorophyll is absorbing the sunlight. Before understanding this thing, we need to understand what is reduction potential. If we understand this word, the complete photosynthesis process becomes incredibly easy to understand. But let's understand what is reduction potential. See, if we understand the word potential is trying to signify the capability. Reduction. This reduction potential of a particular species tries to explain us something. This species wants to reduce itself. What do we understand by the word reduction? Gaining of electron. The species wants to gain an electron. Every single species has a different tendency or affinity towards this electron and wants to gain this electron. And in order to map at how much love they have for the electron, for how much strength and how much intensity they want the electron to come towards that is explained by the reduction potential. In a simple terms, reduction potential, if it has a value in positive, clearly indicates that the species is readily trying to take the electron towards itself. When something is wanting the electron, that means it won't try to give the electron. Let's understand this thing by a particular analogy. See, for a person who is wanting an electron, let's understand that person or a species to be starving. This organism or this species, let's talk about in terms of this chemical constituents, this species is starving. When something is starving, it wants the food, it wants the apple to come towards itself. See, if I'm starving, I want the apple to come to me. I want to find the apple and try to take the apple towards me. So my reduction potential will be high. That means I want to reduce myself. And in case I get an electron, will I be willing to give that electron? No. That means if I say something, that if I have a high reduction potential, that means I want to gain this electron, and in case this electron is being taken from me, I would try to resist it. So if we see this map, in case we are trying to move from one species to another, mapping this, the reduction potential is decreasing. The values here are in the negative. That means highest reduction potential would be seen for this P680 and the lowest would be seen for the species mentioned up. If you understood the thing that I just taught, if reduction potential is high, that means it won't want to give the electron. See, the P680 would not want to donate its electron. It is very starving. It is very starving. Hence, the electron has to be not donated if the will of P680 works. But the situation in the nature is different. The sunlight says, this sunlight comes and brings lot of energy. The sunlight brings lot of energy, and the antenna molecules and the reaction center absorb this sunlight, the energy, and force the P680 to donate an electron to another species. See, P680, which was not wanting to donate the electron, was forced by the sunlight to give the electron to the phaeophyte. This P680 has now given the electron to the phaeophyte. Now this P680 is rapidly starving. It just wants to find any possible direction, any possible source from which it can take back the electron. See, it wants the electron that has been given to it cannot be taken back, hence it sees around itself and finds a water molecule lying here. See, this P680, after being oxidized, that means after donating the electron under the influence of sunlight, tries to find another electron to satisfy its starving. So it uses the oxygen evolving complex and breaks the water, and breaking the water into this species, releasing 2 electrons. This 2 electron goes back to 680 and completes, satisfying its starvation. The P680 is happy again. And what happened here? The H positive was produced. The O2 was produced by the breaking of water. And this breaking of water was somewhat related to sunlight, hence termed as photolysis of water. This photolysis of water led to the evolvement of oxygen as well as H positive as well as electrons, which were given to P680, which is now okay. But the electron which was taken from it earlier went to phaeophyton. This phaeophyton, if we compare this phaeophyton and another species which is termed as plastoquinone, see, phaeophyton lies here, plastoquinone a bit lower. That means plastoquinone is more starving than the phaeophyton. So phaeophyton sees that another person, plastoquinone, is more starving. Let's donate an electron to this. The electron is donated to the plastoquinone. This plastoquinone, after receiving the electron, becomes reduced. The plastoquinone has the electron and follows. After the electron comes to the plastoquinone, it takes the H positive from outside, takes the H positive from the stroma, and after taking the electron and H positive from the stroma, becomes plastoquinol. From the plastoquinone becomes plastoquinol. From a ketone forms a alcohol. This thing turns back into the plastoquinone. See, plastoquinone has accepted the electron, also has the H positive. It wants to turn back into the plastoquinone. This plastoquinone releases this H positive inside, and this electron is returned to plastoquinone. See, the plastoquinone sees the next thing, the cytochrome B6 is more starving. If we follow from the reduction potential scale, is cytochrome BF6 is more starving. The electron which was returned from plastoquinone to plastoquinone is taken forward to the cytochrome BF6. The cytochrome BF6 finds another thing which was the plastocyanin, and then finally plastocyanin was happy to take the electron. Another thing, the P680 from the another thing, the P680 which had given the sunlight has its brother P700. Because the sunlight was falling on P680, similarly sunlight would be falling on P700 now, and this P700 is now forced to give the electron, and it gives the electron to something, the species called X, which has a lesser reduction potential. It is starving again. It says, I need some extra electron, and to its surprise, the neighbor, the plastocyanin, has an extra electron. This extra electron is donated to the P700 and is very happy. See, this P680 had to break down the water, but it does not, because the neighbor, a good neighbor, has the electron. P700, after taking the electron, is satisfied, and before the electron was given to it, it had already lost another electron. This electron moves to species X, then moves to iron sulfur proteins, and this final electron goes to ferredoxin. This ferredoxin is donating the electron to ferredoxin NADP reductase enzyme. See, this ferredoxin donates the electron here. FNR, ferredoxin NADP reductase enzyme. This ferredoxin donated its electron, and using this electron, NADP positive plus H positive was reduced to NADPH. If you would recall, what were the products that we needed after the light reaction? ATP and the NADP. This NADPH is formed, and the H positive, which was pumped from outside to inside, which was obtained from the photolysis of water, are used in the chemoosmotic hypothesis to produce ATP. And this production of ATP, because which was mediated by the sunlight, is termed as photophosphorylation. This is complete non-cyclic photophosphorylation, and in this non-cyclic photophosphorylation we have two products. One is NADPH and another is ATP, which is obtained at this position. If you could say, ATP becomes ATP because of the pumping of electron at this position, pumping of the electron as well as the H positive, the proton. This complete scheme, if seen, follows this pathway. See, if you could relocate, if you could see this in a complete scenario, this goes this way. Isn't it appearing like a Z? See, this appears like a Z. Hence, the non-cyclic photophosphorylation is also termed as Z scheme. From this Z scheme, we had two products. One product was ATP, another product was NADPH. Thank you so much.